All right, so moving into module 3.2, we're going to be talking about fungicides and then also at the end a little bit about nematicides. Now, previously we talked about that, once you have a bacterial disease, that it is highly unlikely that you can overcome that. So we have like resistant varieties or, we might apply an antibiotic just to make sure that we don't run into that problem. And so kind of the same thing about fungi, except that we have different types of fungicides and classifications, activities, modes of action that are going to help us either prevent or if we've already got the fungus or already had the fungal disease, that we can overcome that. We're going to talk a little bit about the basics, kind of modes of action and the mobility within the plant, and then kind of go over a couple of different compounds that are going to, um, they're kind of species specific. So the way that we're going to classify these are going to be like by the distribution, so the mobility within the plant. So this is going to be, is it a systemic? Um, Also, we can classify our fungicides by the chemical group, so the chemical structure. And the reason for this is that these fungicides, these active ingredients, are going to bind to specific places within the plant, uh, like let's say on the cell wall, so that just in case that if that penetration peg does try to get through that cell wall, there is some compound there that is going to prevent that. And then also by MOA, the mode of action or the site of action. You might hear those sort of intertwined there. So we've got the mobility, the chemical group, and the MOA, the mode of action or the site of action. So I've already talked about this several times. We've got contact and we've got systemic, and contact are going to be fungicides that are on the leaf or on the plant, even on the stem, possibly on the roots, and that once that spore lands on the plant, there is some chemical that is likely to disrupt its growth, kind of like a contact insecticide. That we've got this on the plant. When that insect comes into contact with it, it is going to be exposed to that active ingredient, and that active ingredient will then begin to do its thing. It's important to note that chlorothalonil is probably one of the most common contact fungicides that you'll run into. This one, chlorothalonil and mancozeb are probably two of the most common fungicides that you might encounter if you get into a situation where you need a contact herbicide, fungicide. Next, we have systemics, and systemics are going to be absorbed into the plant, and then they're going to be moved throughout the plant by the xylem and the phloem. So that's important that you understand the functionality of those. And then also remember that if you have a bacterial disease and you have this blockage inside of that xylem and phloem, then your fungicide might not be very effective. The important ones to know here are going to be your strobilurins and your triazoles. These are probably two of the most common um, systemic fungicides that you'll encounter. And I really, again, getting back to, I don't expect you to like know all these right off the top of your head. Not many of you are going to be plant pathologists, but just to have that little memory bank in there that says, hey, I remember hearing this. I think I need to look into a strobilurin or a triazole. So just a visualization, we've got our contact fungicide that is on top on our plant, that when that spore lands on that plant, it comes into contact with that fungicide, and it never gets into the plant cells, or if it does get into the plant cells, it does not persist very long because it's being attacked by that fungicide. And then we also have our systemic, in that we're going to spray that on the plant. It is going to absorb into the mesophyll cells. It's going to absorb into the xylem and the phloem, and it is going to move throughout the plant and kind of give us this holistic sort of protection. So here we can see that we have our fungicide on the plant. it is now moving into the mesophyll cells and then making its way into the xylem and the phloem, and then being translocated throughout the entire plant, hopefully throughout the entire plant. And it might be that we apply this systemic fungicide to the roots first, and that we want it to be taken up from the roots because we're expecting to have that sort of a root disease first, but then also that we want to make sure that the xylem and the phloem are protected, and that's probably an easier way to do that. All right, so just to look at a little comparison between the two, and that contact, both of them need to contact and systemic, both of them need to be absorbed. One is immobile, so that once that contact is on the plant, it only penetrates a certain amount. So it's not going to move throughout the plant. It is really just a protective coating. One of the problems is that we don't have much resistance with this contact fungicide, but we can with our systemic, because those fungi strains are going to, they're going to adapt and evolve. So, we've got mancozeb. Again, it's probably one of the ones that you're going to hear the most. We've got our copper sulfate, which is going to be our Bordeaux mixture, and then metalaxyl. Again, I'm not really concerned that like the specific names, because you can always go look that up. All right, so the way that we're going to classify these types of fungicides are going to be preventative, and so this is something that we're going to spray because we expect conditions to be ideal for the potential to be infected, and kind of different from our economic threshold, in that once that, again, remember that once that fungi infect that plant, like that disease can spread very quickly. So especially for our turfgrass people, we're going to go ahead and spray that as a just in case, just because we don't want to lose our turf. Because if we lose our turf, we're going to lose a lot of it very quickly, and it's worth it. From an insecticide standpoint, it might not be worth it to spray it as a preventative. That's a producer decision. Then next we have curative. And so if you do get this infection, this is going to be something that you'll take, that you'll apply to, heal. And then finally we have an eradicant, and this is going to be something that just completely kills everything, all of the spores. And so, if you think about this, this is kind of like taking vitamin C to make sure that your immune system is working, just in case. We're going to take preventative measures to ensure that we don't get sick. If we do, we're likely to go to the doctor and receive either, some antibiotic, or maybe a steroid shot, depending on whatever it is that you get. And then finally we have an eradicant, and this may be something closer to like the flu vaccine, in that you don't ever get that, even though you might still end up getting it, right? So this eradicant isn't necessarily 100% guaranteed that you won't ever get a fungal disease, but we do have these to make sure that, like, just in case if we miss something, that those spores don't continue to proliferate. I put this in here. Maybe, you look back at your notes and you kind of go, okay, these are some of the fungicide classes that I'm going to need. Again, you can really Google all these. All of these different chemical classes, modes of action are going to be in cdms.net, which is going to be in the lab. So again, that's why I'm not really concerned that about mancozeb, or that chlorothalonil, or thiophenate methyl, and have an understanding of this. Now, if we had like a plant fungal class, or if you were in turf management and there was a turf diseases, and when I took this, we did have to know all these different ones. And again, most of the ones that are going to be in green are the ones that you're likely to use. So we've got our, dithiocarbamates, chloronitriles, benzimidazoles, the azoles, triazoles, strobilurins, and then the CAA fungicides. All right, so next is like, how does this actually kill it? What is the mode of action? Fungicides are more metabolic inhibitors, in that they are going to stop the growth of that fungicide. Of that fungus. fungi by disrupting a couple of things. And so we classify those into four broad groups. So we've got inhibitors of, and we'll just talk about the next four things, these inhibitors of this electron transport chain. And so you may remember this from plant science and that photosynthesis, we have the electron transport chain. All organisms have an electron transport chain. That's how they get their energy moved throughout the plant. Throughout their body. Even us, we have an electron transport chain. ATP, mitochondria, powerhouse of the cell, etc. Next, we can inhibit enzymes, and there are certain enzymes that are necessary in order for these plants, in order for the fungi to function. And so if we can disrupt those enzymatic pathways, then that fungi doesn't necessarily grow very well. And if it doesn't grow very well, then the plant can likely overcome an infection. Nucleic acid metabolism and protein synthesis. Imagine if you never ate protein. How strong would you be? Not very strong. So if we can disrupt some of these metabolic processes, then that fungi doesn't grow. And then next, sterol synthesis. And, sterol synthesis is going to be something that goes along with protein synthesis. Just that these four things are what we're likely disrupting when we apply these fungicides in order to minimize the growth of that fungus. So just a different look at some of these. We've got mitochondrial function. Here's our strobilurins. And remember that the mitochondria is the powerhouse of the cell. And so if you don't have any energy being produced by the mitochondria, then you don't have any energy to make proteins, or to continue to grow, to infect the cells, to we've got that intracellular growth. We've got our cell wall function. And if you have, the cell wall is broken down of that fungi, then that means that all of this liquid, all the cytoplasm inside of that, will end up being, oozed out. Proteins and amino acids. And so here's our chlorothalonil, and then sterol biosynthesis. And so this is all of our triazoles. We have multi-site inhibitors where we're going to infect things on the inside of the cell. And then we also have these protectants on the outside, so that those penetration pegs cannot disrupt that plant tissue. They cannot penetrate inside of those cells. And if they can't penetrate inside the cells, then they can't do their job, and they can't get that energy out of that cell. Like, that's their whole goal. The fungi are infecting the plant because there are nutrients and minerals and things that are necessary, like there's energy inside of the plant cell that they need. They aren't able to produce their own energy like that. So they must attack something else. That systemics are going to penetrate inside the plant tissue, that just in case if they do get inside. So we've got a protectant, we've got our protectant coat. And just in case if for whatever reason we have a strain that is strong enough to break through these compounds and it does not affect them, then inside of it we've also got some activity that if that penetration peg does make it in, that we're still disrupting that fungi. So as I mentioned, we do have problems with fungi evolving and becoming resistant. And if we continue to spray the same fungicide over and over again, the exact same mode of action, that eventually the fungi will develop resistance to it. So there is a FRAC code. The Fungicide Resistance Action Committee came up with a FRAC code in that these different chemical classes, as they relate to how, what is the potential for the resistance. And so, our benzimidazoles, and again, I'm not asking you, what is the resistance risk of a FRAC code 11 strobilurin, but you can see that it's pretty high. So this FRAC code may come in handy in that if you spray benzimidazoles 1 year, you probably shouldn't spray it in year 2. You need to find a different chemical class in order to spray that, in order to, to mix it up. You don't want to continue to spray the same mode of action over and over again because those fungi will develop resistance. We've got premixes, and premixes are going to be where we might have more than one mode of action in the same uh, uh, sort of compound, whether that be a dry flowable or whether that be a liquid fungicide. They've got different modes of actions and different chemicals in there that are chemical classes that are going to kind of cover all of your bases just in case. And it might be that they uh, protect against more than one particular fungi. All right, so let's look back at this. We've got our protective, we've got our curative, and we've got our eradicative. And you can see that chlorothalonil, which is a contact herbicide, which is a contact fungicide, doesn't have very much curative power. Now, strobilurins do, and our triazoles do. Now, our premixes are starting to move towards the end where we're getting close to this eradicative, and that we're pretty much taking care and covering all of our bases. All right, so for the quiz, you'll probably need to pay attention to these because I'm going to ask you to at least have some knowledge to remember, or at least be exposed to a couple of these compounds that might, that are going to um, be used for these specific species. And remember, back from like module 3.1, when we talked about the different fungi, these chemical classes are family specific. So once you have your disease and you get it identified and you know that it's an Oomycetes, then you know that you're likely to use some of these particular compounds. You identify that you have a powdery mildew, you verify that it is Ascomycetes, and you are going to spray mancozeb or pyraclostrobin or maybe even thiophenate metal. And then for like our alternaria or our rhizoctonia, which is super, rhizoctonia is very detrimental to turf grass. And so you'll want to have some triazoles on hand. Big picture takeaway is that you need to have several different modes of action depending on, like, so that way, just in case if you do get one of these different diseases, that you've got something on hand that can help you overcome that. I did mention that our strobilurins are going to be a popular one in that, they don't have very much risk to humans or the environment, and they're also going to be very effective in towards not only the curative but also the preventative stages of um, prevention, like prevention and making sure that brain too early, making sure that you can overcome those. All right, and so there are going to be different chemical structures. Once they found out that they've isolated these from these mushrooms, they can then synthetically make these compounds, and each different company is going to have their own compound that is going to affect something, some way, shape, or form. But at some point in time, this was like 1992. Most of these chemical compounds have probably come off patent, and so now anyone can make these. And so this is important because you might not necessarily want to buy that, or you need to buy that from Syngenta, and other companies can make that. It's kind of like Roundup is known as glyphosate, but there are other companies that are making that as well. Also, there are quinone outside inhibitors, QOI fungicides, and they're going to cover a broad spectrum. So these are good to have on hand and just so that you can, spray that across your crops or your turf, and you're likely to get a more robust protection against different diseases. It's important also to understand that there's going to be some crop compatibility, and this will be on the label. So, like, to make sure that you're not spraying this on something that you shouldn't, especially something that's going to be, uh, uh, well, depending on the rate, and then also that these different crops need to have these receptors on their cells somewhere, and they've done a lot of research on that. So again, these are going to be where they're going to move into the plant. They're going to move um, into the vascular system. So we've got our contact, we're going to move into the mesophyll cells, make our way into the leaf and hopefully into the vascular system, and that can be translocated throughout the plant, and we get this overall protection of that. It's important for this mobility, like the compounds need to be able to move through the plant, especially if you're trying to get full plant coverage, right? So there's one thing about having that just around the edges, or on the surface of the plant versus inside. You'd rather have that inside of the plant. So QOI fungicides, you probably want to apply those before the fungi develop. And again, getting back to this, like, it's worth it to spray a preventative a fungicide application, so you don't get the disease. Because if you get the disease, you might not be able to catch it in time. Here we're just looking at how these different fungicides actually work. We've got our spore germination. That germ tube is going to move in, and then that mycelial growth is going to take over that. And these QOI inhibitors actually protect across all of these different stages. Strobilurins are going to be that mitochondrial respiration, in that the mitochondria is the powerhouse of the cell, and if it's not producing energy, then the fungi isn't growing. If the fungi doesn't grow, then the plant can overcome that. I think it's more important that you know about this than you know about these cytochrome Bs and As and C1s. If you move into that area, if for whatever reason you move into plant pathology, this would be something that you would probably be more familiar with, just to understand how that fungicide is working. For us, we don't know when we're going to encounter this, and so having an understanding that there are different fungicide classes and those 4 ways of inhibition, and then using cdms.net, to me personally, and to give to you, is more important that you understand the actual mechanisms of how this fungicide works. All right, so summing this all up, we've got resistance management, and the fungicides can be they can become obsolete pretty quickly, because fungi are going to adapt and grow uh, to overcome this. So, you want to limit the number of applications. You don't want to use consecutive applications of the same mode of action. We've got premixes that are going to give you a larger coverage area, and also, potentially against different strains of fungi. We would prefer to spray those as preventative or protective. And then you can also tank mix other fungicides with these, so that maybe you don't want to buy the premix, and you have let's say that you know that you're more susceptible to a different type, like one specific type of fungi, and you need to spray a lot more of that fungicide to prevent that. But you can also add some other ends as a preventative measure. All right, so that's going to end the fungicides. We're going to move into nematicides next. And just again, quickly, I just want to make sure that you have at least been exposed to some of this information, and that you have an understanding of what is actually happening for our nematodes. So, nematicide, or ICIDEs, are going to be something that kills a plant parasitic nematode. But when I've been looking through this information, what I found is that they're really more nemastats, or I'm sorry, nematostats. And they don't necessarily kill the nematode directly, as like we would think about like wasp spray, where if you spray that wasp dies pretty quickly. But what ends up happening is that there are these compounds that either paralyze or they disrupt the feeding mechanisms or the ability to ingest the compound, or maybe there's like some allelopathic uh, um, property that is going to sort of deter that nematode from eating on that plant root. So we've got natural and artificial, and our artificial are going to be our fumigants, carbamates, and organophosphates. And so if you remember back from our insecticides, these are also used for insects, but these are probably going to be added as a drench into the soil. Just a look to see that, like, these compounds do have some benefit in that here's our untreated. This is a known infected area of, with nematodes, and we have these different compounds that are tend to, seem to show some promise, and that you can see how stunted these plants are compared to how full these are. All right, so our natural ones, there are going to be some, garlic polysulfide products. We've got neem cakes. We've got root exudates of marigold. So, kind of allelopathy. That's kind of Dr. Pierce's area of expertise, or at least knowledge base. And then we've got those. We do have some fungi that are predators of nematodes. Nematodes. So there's going to be a biological control for that. We got these neem cakes, and these are compounds that are, we kind of got these cakes. These will dissolve and then deliver that into the soil that is going to kind of protect our plant roots and sort of fend off, or sort of, be just a disruptive environment for those nematodes. You might remember this from soils, and I showed this as a slide that this fungi is now going to pretty much consume this whole entire nematode because it contains the energy that it's needing. And we prefer to take over our nematodes than our plants. Artificial nematicides, fumigants, carbamates, and organophosphates. And I really want to talk about the fumigants first because once they were spraying these back in the 60s and the 70s, the 80s, and some of the 90s, and then they realized that these fumigants were causing more problems than they were worth, and we have now sort of transitioned into carbamates and organophosphates. But it's important that you've at least been exposed to this particular at least a couple of these compounds, just so that you're aware that you're probably not supposed to use these. And those compounds are at least methyl bromide. I at least want you to be aware of methyl bromide, chloropicrin, and metam sodium. These are three of the most common compounds that were used to combat nematodes, because they're microscopic worms, and so you really got to figure out a way in order to get some contact in there with them. So we'll discuss these just a little bit, just so that you have some knowledge of that. This DD is a mixture of 1,2 and 1,3 dichloropropane, propane, whichever. They found out that it was causing groundwater contamination, and of course, once it causes some contamination of groundwater, they're going to end up stopping that. So now we have this issue where we can't use this D-D or DD2D, whatever it's called. And they had to come up with something new. Previously, we talked about that this 1,2 dichloropropane probably wasn't important, um, but they changed the formulation to 1,3 dichloropropane, probably not needing to know, but they did find that there was some inactivity for a specific uh, species of nematodes. The ethylene dibromide, they found out that this was causing male sterility, and so they stopped using that as well. Again, these are fumigants. Methyl bromide, though, methyl bromide is probably one of the most familiar ones. It says here that it's the fourth most commonly used pesticide. Here's the thing about methyl bromide. And the way that they do this is they'll cover it with a tarp, and then they'll spray that fumigant underneath the tarp. Think about it like a gas. But what that does is that kills everything. It kills everything in the soil. And so if you use methyl bromide, then that means that you have to add back all of the bacterial populations, all the fungal populations, and we've talked about that our insect populations are important as well. Our weed populations not so much, so it was good for that, and for the nematodes, but there are going to be some drawbacks to that. And so somewhere in the early 2000s they realized that, like, hey, this methyl bromide is ozone depleting, and we don't need to be using that. So methyl bromide is definitely one that you should have at least heard coming out of an agricultural program as a fumigant for nematodes. Chloropicrin is one of the oldest soil fumigants. It has both herbicidal and nematode activity, but it's toxic. I'm not real familiar with this one, but it seems that once they realized that most of these compounds have this ozone depleting potential, they kind of went away from that. I mean, these are very difficult to apply. Next is metam sodium, and metam sodium eventually gets converted to this methyl isothiocyanate, or MITC, and that is actually what kills or actually works on the nematodes. So it also has, the potential to kill all of these things. But again, when you're applying this fumigant and you're basically sterilizing the soil, you have to add all of that microbial population back. This sodium tetrathiocarbonate is going to get converted into carbon sulfide, disulfide, and that is what's going to act on that nematode. So we're applying these compounds that need to be broken down or converted into a secondary metabolite, and that is what is the actual active compound for eradicating these nematodes. All right, so I talked about that. It is not likely that some of these compounds are not actually specifically intentionally killing the nematode, but there is some interaction with that nematode that is causing a disruption where that nematode cannot eat on that plant root. So this fumigant is going to penetrate that nematode is that fumigant is going to penetrate the body wall, so it doesn't have to be consumed by the nematode. And then it all, like, essentially what happens is that there's going to be this bioaccumulation of that compound, and that is what's going to end up disrupting the organs. And so think about if your liver stopped working, kind of the same principle, or if your gut wasn't working for this nematode, then you won't be able to consume the nutrients out of the root. This metam sodium, again, it has to be converted into this secondary byproduct, and it's going to disrupt this enzymatic nervous or respiratory system. So very similar to what happens with our insecticides, in that they're going to penetrate the insect, or in this case, the nematode, and it's going to disrupt some metabolic function. I think broad picture, it's just going to disrupt something. And if it's disrupted, then that means that the insect, the fungi, the nematode, the bacteria, whatever it is, isn't operating at full capacity. So this metam sodium's pretty quick. It can work pretty quick. Um, what happens is again is that this metam sodium gets converted into MIT-C. That MIT-C gets absorbed into the cell walls and the body, and it disrupts these enzymatic nervous and respiratory systems because it bioaccumulates. So you don't necessarily have to apply a lot, but it's going to be easily absorbed by the nematode. And as it gets absorbed, it is going to disrupt those bodily functions. All right, real quick, just to get into the carbamates, in that, remember we have our chemical name, our common name, and then we have our trade name. And so, if you are going in looking for something to um, apply for your nematodes, you'll ask for an aldoxycarb, and you can probably Google this, or you can put that into cdms.net, and it'll tell you what the compound is. So I know I've said this several times. CDMS.net is where you're going to go to find the information that you need. You don't have to memorize these compounds. And if you get into the field, the more you use them, the more you'll be familiar with those different compounds. And then carbofuran. I probably won't be able to find this because they found out that it kills the birds. And so if we're going to kill things other than the nematodes, or if we're going to, be contaminating groundwater, then probably not going to be able to get this. And it would seem that you would probably need a pesticide applicator's license in order to get these compounds, because they're going to be pretty stout. Oxamil, this one is going to control a specific um, type of nematode. This is probably what you'll get compared to this aldicarb. And so, it's probably more widely used, and it's not as toxic or persistent in the soil as an aldicarb. So this is probably going to do its job and then be broken down pretty quickly. Now, this one also has systemic activity, so you can apply this to the plant and it will make its way to the roots. This is one of these aldicarbs are one that is not necessarily specifically killing like the nematode specifically, but the death does occur because of disorientation or starvation. It's really hard to think about that this microscopic worm is disoriented in the microscopic world, but it's unable to control itself. And so you might remember that I talked about that. Maybe this is in the next slide or two. In the organophosphates, the guy that was the pesticide applicator teacher for me, and he had that shake that when he wrote, he wasn't able to control himself. And so, this is going to, once it's been translocated to the root, it's going to inhibit the feeding. And so there's going to be this sort of smell or barrier that sort of protects the roots. And so the nematode doesn't want to interact with that compound. And if they do, then they'll, be disoriented and likely starve. Next, we have our organophosphates. We got ethoprop. You can see this has started being used in the 1960s, and that, at least it's non-carcinogenic, so they're probably still using this. Venamifos, again used in the 60s. It's acutely toxic, but also not shown to be. So instead of your carbamates, you're likely going to go look for an organophosphate. Like this one, we like caducephos, caducephos. in that it's not going to contaminate the groundwater. So it would seem that organophosphates is likely to be your choice compared to fumigants or carbamates. Next, we have phosphothioate, a little bit newer, broad spectrum activity. We're controlling these lesion and root knot nematodes. And so here again, we have our mode of action for our organophosphates is going to be that acetylcholinesterase inhibitor. This is the shaking in that the nervous system does not have control over the body. And so not only are we spraying these for our insects, but we're also applying them for nematodes. So a little kind of dual action there. Just to be aware of, if you have some nematodes, you're probably going to go look for organophosphates. Abnormal behavior, paralysis, and ultimately death. So they're probably more for insects, but they also do have some, some narcotic effect and behavioral modification. We're going to disrupt those nerve impulses at high concentrations. They pretty much just shake themselves to death. We can have delayed egg hatch and molting, and if we don't molt properly, then we're going to have some, body development. Our plant tissues, and so. If we have this delayed hatch and molting, then that means that there's not going to be any development in the plant tissues, and that's going to reduce those nematode effect, infection, development, and reproduction. So we're not only going to spray these for insects, but we're also going to get some, nematicide or at least some nematistat, because we're not actually specifically killing nematodes. We're actually disrupting their behaviors and their ability to feed on those roots and those plants, and they die from starvation. We got a couple of new ones that have come out, it seems. This Nematicides is one. Within about 1 hour of contact, we're going to cease feeding and they become paralyzed. And within 24 to 48 hours, they're pretty much dead. Then we also have MCW2. This is going to be, for our root knot nematodes. And again, I just want you to have at least heard these and seen them, just in case if you get into the realm, you at least know how to find them. That's why I think it's important to understand how to use cdms.net more than it is about remembering MCW2 and what it does and doesn't work on. All right, so that ends it for module 3.2. The lab is going to go over using cdms.net in order to find out different chemicals that you can use for fungicides, insecticides, and also our nematicides. All right.